Lymphatic system. So what is lymphatic system? Lymphatic system is a circulatory system that made up of lymph vessels that drains extra fluid that has passed out of the blood into tissues. So we can say lymphatic system represent an accessory route through which fluid can flow from the interstitial space into the blood. The lymphatic system is very much important as it can carry proteins and large particulates from the tissue space to the blood, which cannot be absorbed directly by the blood capillaries. This return of protein to the blood from the interstitial space is essential, without which we would die within about 24 hours. Formation of lymph. Lymph is derived from interstitial fluid that flows into the lymphatics. Therefore, lymph, as it enters the terminal lymphatics, that has the same composition as that of interstitial fluid. Therefore, lymph is composed primarily of interstitial fluid with variable amounts of lymphocyte, bacteria, cellular debris, plasma proteins, and other cells. In our most of the tissues, the average protein concentration of the interstitial fluid is about 2 gram per dl. Therefore, the protein concentration of the lymph that flowing these tissues also will be the 2 gram per dl, but the lymph that formed in the liver has a protein concentration of as high as 6 gram per dl. Lymph formed in the intestine has a protein concentration as high as 3 to 4 gram per dl. About 2/3 of the lymph normally is derived from the liver and the intestine, and 1/3 from the rest of the body. Thus, the thoracic duct lymph, which is the mixture of lymph from all the areas of the body, usually has a protein concentration of 3 to 5 gram per dl. Lymphatic system is one of the major route for absorption of nutrients from the GI tract, especially for the absorption of fats in the food. So after a fatty meal, the thoracic duct lymph sometimes contain as much as 1 to 2% of fat. Sometimes larger particles such as bacteria can get enter into the lymphatic capillaries by pushing themselves in between the endothelial cells of the lymphatic capillaries. Thus, they enter into the lymph, and then the lymph passes into the lymph nodes, and these particles are almost entirely removed and destroyed inside the lymph node. Almost all the tissues of the body have special lymphatic channels that drain the excess fluid directly from the interstitial space. But there are some exceptions, which are superficial portion of the skin, the central nervous system, the endomysium of the muscle, and the bones. Even these tissues have minute interstitial channels. These are called the prelymphatics, through which interstitial fluid can flow. This fluid eventually empties either into the lymphatic vessels or, in case of brain, into the cerebrospinal fluid, then directly back into the blood. So these tissues we just mentioned for the exceptions, the interstitial fluid from those tissues are also being transferred into the lymphatic vessels and then into the blood. Essentially, all lymph vessels from the lower part of the body eventually empty into the thoracic duct, and then it empty into the blood venous system at the junction of left internal jugular vein and the left subclavian vein. Lymph from the left side of the head, left arm, and the left side of the chest also drain into the thoracic duct before it empty into the vein. That means at the juncture of left internal jugular vein and the subclavian vein. The lymph from the right side of the neck, head, and the right arm and the part of the right thorax enter the right lymphatic duct. This right lymphatic duct is much smaller than the thoracic duct. This right lymphatic duct empties into the blood venous system at the juncture of right subclavian vein and the internal jugular vein. So the lymph from the right side of the neck, head, right arm, and the part of the right thorax only empty into the right lymphatic duct. And the rest of the body, that means the lower portion of the body and the left side of the head, left arm, and the part of the left side of the chest, all drain into the thoracic duct. Terminal lymphatic capillaries and their permeability. The most of the fluid filtering from the arterial end of the blood capillaries reabsorbed back into the venous end of the blood capillaries, but on average about 1/10 of the fluid enters into the lymphatic capillaries and return to the blood through the lymphatic system rather than through the venous capillaries. So the blood that filtered out from the arterial end of the blood capillaries, that fluid flows among the cells, finally get reabsorbed back into the venous end of the blood capillaries. But not all the fluid get reabsorbed at the venous end of the blood capillaries. About the 1/10 of the fluid enters into the lymphatic system and then it returns back to the blood through the lymphatic system. The total quantity of all this lymph is normally only 2 to 3 liters per day. The fluid that returns to the circulation by the way of the lymphatics is extremely important because substance of high molecular weight such as the protein can enter into the lymphatic capillaries almost unimpeded, although the substance of this high molecular weight protein cannot be absorbed from the tissues in any other way. This is due to the special structure of the lymphatic capillaries. These proteins are very much essential to maintain the blood oncotic pressure, and it also has so many important functions. So these are essential for life. The reason for this mechanism is the special structure of the lymphatic capillaries. We can see from the picture that the endothelial cells of the lymphatic capillary attach to the anchoring filament to the surrounding connective tissue. The endothelial cells of the lymphatic capillary are connected with the surrounding connective tissue. At the junction of the adjacent endothelial cells, the edge of the one endothelial cells overlap the edge of the adjacent cells, such a way that overlapping edge is free to flap inwards. Thus it forms a minute valve that opens to the interior of the lymphatic capillary. So these endothelial cells of the lymphatic capillary are anchoring to the surrounding connective tissue. So the edge of the one endothelial cell is overlapping the adjacent endothelial cell in a such a way that it can freely flap inwards, just forming a valve that open inside. Interstitial fluid along with its suspended particles can push the valve and flow directly into the lymphatic capillary. However, this fluid has difficulty leaving the capillary once it has entered. Once it has entered, that cannot move out from the lymphatics easily. Endothelial cells of the lymphatic capillaries are arranged such a way that when the fluid tries to go out from the lymphatic vessels, it causes closing of this flap valve. Thus, the lymphatics have valves at the tip of the terminal lymphatic capillaries as well as along their larger vessels up to the point where they empty into the blood circulation. Rate of lymph flow. About the 100 ml per hour of lymph flows through the thoracic duct at a resting human, and approximately another 20 ml flows into the circulation each hour through the other channels. So the total estimated lymph flow is 100 ml, which is in the thoracic duct, and 20 ml through the other channel. So total is the 120 ml per hour, or if we calculate in a day, that would be 2 to 3 liter per day. Effect of interstitial fluid pressure on lymph flow. The normal interstitial fluid pressure is -6 mm of Hg. When the pressure of the interstitial fluid is more negative than this value, the normal lymph flow in this situation is very little. When this pressure rises to 0 mm of Hg, that means at the atmospheric pressure, the flow increases more than 20 folds. Therefore, any factor that increases the interstitial fluid pressure also increases the lymph flow if the lymph vessels are functionally normal. The factors that increases the interstitial fluid pressure are elevated capillary hydrostatic pressure, decreased plasma colloidal oncotic pressure, increased interstitial fluid oncotic pressure, and increased permeability of the capillaries. All these factors causes a balance of fluid exchange at the blood capillary membrane to favor fluid movement into the interstitium, thus increasing the interstitial fluid volume, and that increases the interstitial fluid pressure, and also that increases the lymph flow. However, when the interstitial fluid pressure becomes 1 to 2 mm of Hg, that means when the pressure is greater than the atmospheric pressure, the lymph flow fails to rise any further. This results from the fact that increasing the tissue pressure not only increases the entry of the fluid into the lymphatic capillaries, but also compresses the outside surface of the larger lymphatics. That means when there is increase in the pressure obviously increases the lymph flow, by increasing the interstitial pressure. But this increased pressure also causes compression of the surface of the larger lymphatics. So when there is a pressure from the outside, that will obviously impede the lymph flow. At the higher pressure, these two factors, one pressure is the increased interstitial pressure that causes the increase in the lymph flow, and the other pressure is the pressure on the surface of the lymphatic vessels that impedes the lymph flow. At higher pressure, these two factors balanced each other, so the lymph flow reaches to a maximum flow rate. Lymphatic pump increases the lymph flow. There are valves at the lymph channel. It is shown that collecting lymphatics or the large lymphatic vessels become stretched with fluid, and the smooth muscles in the wall of the vessels automatically contract. Furthermore, each segment of the lymph vessel between the successive valves function as a separate automatic pump. So the segment of the lymphatics between the two valves, upside valve and the lower side valve, this is the segment. The segment between these two valves acts like a separate automatic pump. When there is slight filling in this segment, that means in between the two valves, and there is slight filling occur, this stretches the vessels, so the vessel wall automatically contracts. So when there is contraction, the fluid is pumped through the next valve. So fluid is pumped through the upper valve and lymph passes to the upper segment or the next segment. And in the next segment, when there is filling, that also causes stretching of the lymph vessels. That leads to the contraction of the smooth muscles on the wall of the lymphatics of that segment. Thus, fluid fills the subsequent segments. This process continues all along the lymph vessels until the fluid is finally emptied into the blood circulation. In a very large lymph vessels such as thoracic duct, the lymph pump can generate the pressure as high as 50 to 100 mm of Hg. Pumping caused by external intermittent compression of the lymphatics. In addition to the pumping caused by the intrinsic intermittent contraction of the lymph vessels wall, any external factor that intermittently compresses the lymph vessels can also cause pumping. As we just learned that when there is collection of some fluid into the lymph vessels, that causes the stretching of the vessels, and this leads to contraction of the smooth muscles of the wall. This leads to pumping. This is called the intrinsic intermittent contraction. But if there is some factors that intermittently compresses the lymph vessels from the outside, that can also causes the pumping. Such factors are as follow: contraction of the surrounding skeletal muscle, movement of the part of the body, pulsation of arteries adjacent to the lymphatics, compression of the tissues by objects outside the body. The lymphatic pump becomes very active during exercise, often increases the lymph flow to 10 to 30 folds. But during the periods of rest, the lymph flow is sluggish, that is almost zero. Lymphatic capillary pump. The terminal lymphatic capillary is also capable of pumping lymph. As we have noted, the wall of the lymphatic capillary are tightly adherent to the surrounding tissue cells by means of their anchoring filament. Therefore, each time excess fluid enters the tissue, that causes tissue to swell, so the anchoring filaments pull on the wall of the lymphatic capillaries. As the anchoring filaments are attached to the tissues and the tissue gets swell, that obviously pulls the anchoring filament, so that increases the gap between the adjacent endothelial cells of these lymphatic vessels. As there is gap increase, so that allows more amount of the fluid to flows into the terminal lymphatic capillary. And then when the tissue is compressed, the pressure inside the capillary increases as the fluid already has entered inside the capillary, and the edge of the endothelial cells of the capillary are overlapped in such a fashion that when the fluid inside the lymphatics tries to come out from the lymphatics, that overlaps each other. The overlapping edges of the endothelial cells close like a valve. Therefore, the pressure pushes the lymph forward into the collecting lymphatic instead of backward through the cell junction. The pressure which is built inside, that pushes the lymph forward into the collecting lymphatics. This lymphatic capillary endothelial cell also contain a few contractile actomycin filament. These filaments have been observed to cause rhythmical contraction of the lymphatic capillaries in the same rhythmic way that many of the small blood vessels and the large lymphatic vessels contract. Therefore, it is probable that at least part of the lymph pumping result from the lymph capillary endothelial cell contraction, in addition to contraction of the large muscular lymphatics. So the summary of the factors that determine the lymph flow are: from the previous discussion, now we can see that the two primary factors that determine the lymph flow. Number one, the interstitial fluid pressure, and number two, the activity of the lymphatic pump. Therefore, one can state that the rate of the lymph flow is determined by the product of interstitial fluid pressure times the activity of the lymphatic pump. The lymphatic system plays a key role in controlling interstitial fluid protein concentration, volume, and pressure. It is already clear that the lymphatic system functions as an overflow mechanism to return excess protein and excess fluid volume from the tissue space to the circulation. Therefore, the lymphatic system also plays a central role in controlling the concentration of protein in the interstitial fluid, the volume of the interstitial fluid, and the interstitial fluid pressure. Small amount of the proteins leak continuously out of the blood capillaries into the interstitium. Only a minute amount of the leaked protein return to the circulation by the way of venous end of the blood capillary. Therefore, these proteins tends to accumulate into the interstitial fluid, which is in turn increases the colloid osmotic pressure of the interstitial fluid. The increasing colloid osmotic pressure in the interstitial fluid shifts the balance of force at the blood capillary membrane in favor of fluid filtration into the interstitium. Therefore, fluid is translocated osmotically outwards through the capillary wall by the protein and into the interstitium, thus increasing both the interstitial fluid volume and the interstitial fluid pressure. That means, normally, small amount of the protein comes out from the blood capillary into the interstitium. As the protein accumulates inside the interstitium, that increases the osmotic pressure. As the osmotic pressure increases, it attracts water from the blood vessels. This leads to increase in the fluid volume of the interstitium. As the fluid volume increases and also the number of the protein increases into the interstitial fluid, that increases the pressure of the interstitial fluid. The increasing interstitial fluid pressure greatly increases the rate of the lymph flow, which carries away the excess interstitial fluid volume and excess protein that has accumulated into the space. As the rate of the lymph flow is directly proportionate to the fluid pressure at the interstitium, so when there is increased interstitial fluid pressure, that increases the rate of lymph flow. So the excess interstitial fluid and the excess protein that has accumulated in the interstitium is carried away from the interstitium into the blood vessels. Therefore, once the interstitial fluid protein concentration reaches a certain level, so when the some proteins comes out from the capillary end of the blood vessels into the interstitium, and this protein continuously added up, as there is increased protein, that increases the osmotic pressure, that attracts water, so ultimately that increases the interstitial fluid and the pressure, that increases the lymph flow via the lymphatic system. So ultimately it comes to a balance when there is protein leak from the blood capillaries equilibrate with the protein absorption via the lymphatics. Therefore, the quantitative values of all these factors reach a steady state, and they will remain balanced at this steady state level until something changes the rate of the leak of the protein and the fluid from the blood capillaries. Unless and until there is any changes in the rate of the leak of the protein or the fluid from the blood capillaries, the quantitative values of these factors, of all these factors, reaches a steady state, and they will remain balanced in this steady state. Significance of negative interstitial fluid pressure as a means of holding the body tissues together. Traditionally, it has been assumed that different tissues of the body are held together entirely by the connective tissue fibers. However, connective tissue fibers are very weak or even absent in many places of the body, particularly at points where tissues slide over one another. For example, skin sliding over the back of the hand or over the face. Yet, even at these places, the tissues are held together by the negative interstitial fluid pressure, which is actually a partial vacuum. When the tissues lose their negative pressure, fluid accumulates in the spaces, and the condition is known as edema.